Hello and welcome everyone to this podcast for Epigenetics. Today we're going to talk about cancer and its link to epigenetics. But we got to begin with a brief background of cancer and what cancer is before we jump into epigenetics. I want to begin by talking about some general information on cancer. Even though our understanding of cancer has improved dramatically over the decades, and the treatments have improved in such a way that the chances of surviving cancer has increased over the years. However, there are still approximately 1.6 million new cases each year in the US alone. There is also approximately 600,000 deaths from cancer each year, also in the US. When we look at the current incidence rates of cancer, it's estimated that people born between 2007 and 2009, that approximately 41% of those individuals will get cancer. So clearly there's a lot more work to be done. Even though the chances of surviving cancer have improved, these numbers speak to the fact that it's still an incredibly detrimental disease. And it's incredibly likely that everyone listening to this podcast has had a family member or a friend directly or indirectly affected by cancer. So let's define cancer next. Now, cancer is hard to define. And it's hard to define because there are over 100 different types of cancer. And because there are over 100 different types of cancers, it's like having 100 different types of diseases. And it's unlikely that there will be a single cure for all types of cancer. But we can define cancers as having some general features. The first I'm going to call tumor formation. This results from an overproduction of abnormal cells. We'll talk later about how they become abnormal cells. That results in a mass of cells developing. These tumors, as they're being formed, can impact organ function. They don't necessarily have to, but they often do. Now, often at this stage, we can remove the tumor, and that can lead to the normal functioning of that organ. And as long as the tumor hasn't hit this next stage, which we'll talk about in a moment, we don't think of cancer as having set in. For cancer to firmly be established, this next stage of metastasis must occur. This is when cells from the tumor migrate to other tissues or organs and grow a new tumor. When this happens, we say that the tumor has metastasized. At this point, the tumor is malignant. And at this stage, we say it is cancer. But let's first talk about how the tumor will form. I want to mention this term called homeostasis. Homeostasis can be defined as the tendency to move towards a physiological, stable equilibrium. So to think about homeostasis, let's think about how an organ like the liver can be maintained. And I'm going to draw the liver like so. Very jealous, I'm sure, of my ability to draw a liver. Now this liver could get smaller, or under some circumstances it could get bigger. It could get smaller because of cell death, either apoptosis or necrotic death, through injury, some kind of trauma, but the end result is a smaller liver. Well, this is balanced with the formation of new cells, either from stem cells in the liver that need to be activated, or it could be from already derived cells that just need to go through the cell cycle to produce new liver cells. But the end result is maintaining this liver of the normal size. Now, the liver can also get larger over time. These are often environmental related causes when it gets larger like this. For instance, the drug phenobarbital will cause the liver to get much larger. But it's balanced. I should have drawn an arrow in reverse on this side. So back over here, the larger liver has to be balanced by cell death. So for instance, when phenobarbital is added, the liver gets larger. When it's removed, cell death occurs and it goes back to a normal shaped liver. In fact, the liver has such a great capacity to regenerate. We can remove 90% of the liver and it will grow back over time. This, of course, is done experimentally. I wouldn't advise you to lose 90% of your liver. This is just to demonstrate the power of homeostasis, that the tendency to maintain this normal-sized liver is accomplished through homeostatic events. Let's go back to cancer. We can think of cancer as the result when this balance is upset. That is, when more growth occurs and less cell death occurs. This can be caused by one of three mechanisms. The first is when apoptotic mechanisms fail. So usually cell death or apoptosis will keep a large liver in check. But when those mechanisms fail, then growth predominates. The next is when cell division regulation fails. Usually the cell will only divide, that is, get larger, which allows the whole organ to get larger. It will only divide when it's signaled to do so. And there are regulation mechanisms involved to make sure that's the only time it happens. But sometimes these fail, and that will lead to excessive growth in the organ. We can also see this when we have failed regulation of stem cell activation. Within organs, there are usually stem cells, and these stem cells are responsible for making new cells. If the regulation of these stem cells fails, they may produce more and more and more cells, even though they're not supposed to be making more and more cells. So now let's think about this second point in a little bit more detail. How is the cell division regulation maintained, and what causes it to fail? So let me draw the cell cycle here. We have G1, which is followed by S phase, which is followed by G2, which is followed by M phase. G1 and G2 both stand for gap. At G1, the environment is evaluated. That is, are the cells receiving signals for division? It also checks the DNA to make sure there are no mutations. If there are mutations, the cell will either fix those mutations or this cell will enter into apoptosis. To make sure that happens, there is a G1S checkpoint. During S phase, once it's past this G1S checkpoint, during G2, DNA replication is verified. That is, has all the DNA been copied? Next it'll check are there DNA mutations? If there are, it will either fix those mutations or send the cell for apoptosis. If there are too many mutations, it's better to have the cell enter apoptosis. It also makes sure that cell is ready for M phase. At M phase, the chromosomes are segregated to new cells. To make sure this happens, there is a checkpoint here, which is called the spindle checkpoint. And at the spindle checkpoint, it makes sure that the DNA or the chromosomes are attached to spindles. If they're not attached to the spindles, the chromosomes won't segregate properly. I forgot to mention the name of this checkpoint here. It's cleverly called G2M. So as I said in the beginning, there are many types of cancers, and they are all characterized a little differently. But at the cellular level, the first thing that happens is a cell gains a mutation that allows it to go past a checkpoint. And I'm going to talk about the G1S checkpoint here, just for an example. This checkpoint is heavily regulated by the protein called retinoblastoma. We'll mention it later when it comes up again, but retinoblastoma is a tumor suppressor gene. So under normal circumstances, retinoblastoma will bind to E2F. And E2F, as we'll describe later, is a proto-oncogene. But we'll come back to those terms in a moment. Now, at this checkpoint, the cell is assessing the DNA. Is there damage? Is there no damage? Is it ready to move to S phase? If it's ready to move to S phase, retinoblastoma, the RB protein, will become phosphorylated. And that will allow E2F to be released. And once it's released, it will bind to specific genes because E2F is a transcription factor. It will bind to genes necessary for S phase. So I'm just going to put S phase genes. And when it binds here, it will cause the transcription of these genes to make the proteins. And that will allow it to go to S phase. Now I've really simplified that. There's many other proteins involved in this process. Now this is how it normally works. But for instance, let's say retinoblastoma was mutated in such a way that it was unable to bind to E2F. When that happens, E2F will always be active, will always bind to the promoters, and always turn on these S-phase genes, thereby blowing past this checkpoint and copying the genome regardless if there are mutations or no mutations. Now what happens next is, because you're no longer stopping at this checkpoint, additional mutations arise. These other mutations may affect the telomere, angiogenesis, cell contact regulation. Usually cells will only grow within a certain area and then they will stop growing. So if you had a petri dish here that you were growing cells in, they would usually just form a layer on the bottom. But if they're cancer cells, what will happen is they will start to mound up on each other like so. Some other features of these additional mutations might affect the need for nutrition. Cancer cells typically require less nutrition. Now, usually what happens, luckily, is when these cells begin to go rogue and not behave as they're supposed to, our immune system can often detect those and destroy them, but not always. So some of these mutations could affect the ability to be detected by our immune system. Typically speaking, for a cell to go from its first mutation to full-blown cancer, it requires approximately 10 mutations. Now, every cancer is different, and not every cancer begins this way. This is just one example. Okay, let's go ahead and move on, and I want to talk about the classes of genes that are associated with cancer. There are two classes of genes I want to talk about as they relate to cancer. The first are the proto-oncogenes. And those are the ones that move the cell cycle past these checkpoints. And remember, we had these three checkpoints. We had the G1S, the G2M, and the spindle checkpoint here. Proto-oncogenes are given the signal that the cell is ready to divide, and they initiate the next step of the cell cycle. They are often provided some sort of signal. So if this is our cell here, various signals called mitogens bind these receptors and tell the cell to divide, in which case the proto-oncogenes are activated. Now, in a typical cell, the decision to divide only occurs about 50 times. Now, this depends on the cell type. In general, a eukaryotic cell, a human cell, will divide about 50 times before it is programmed to die through apoptosis. So we can think of proto-oncogenes as the genes that drive the cell cycle forward. An example would be E2F, the one we talked about on the last whiteboard that binds to retinoblastoma. And another one is HER2. And this is an important one. It's strongly linked to breast cancer. So now in cancer, these proto-oncogenes are converted into oncogenes. And oncogenes, they will produce a protein that disobeys, so to speak, these stops. They will go straight through these stops regardless if there are mutations or no mutations in the genome. They will go through the stop here, regardless or not if mutations were created. And same thing here, they'll blow through the spindle checkpoint, regardless if the spindles have been attached. We consider these mutations to be gain of function, or we could call them dominant mutations. And a dominant mutation, if you remember back from genetics, is one that only one of the two alleles needs to be mutated. So remember, in a typical cell, you will have 2 copies of every gene. And we have our proto-oncogene here. I'll just abbreviate it as PO. And I'm going to put an R over here because the activation of this proto-oncogene is highly regulated. And we have 2 copies, and each of these copies is highly regulated. But when they become a proto-oncogene, one of these is no longer regulated. It's just on all the time. The other one may or may not still be a proto-oncogene and regulated. It only takes one of these to be overexpressed in such a way to go through these stop points. So in this case here, this gene will be constitutively on all the time. Now, how do you make an oncogene constitutively active? There are 3 general ways. We can have a point mutation, and this point mutation would produce a protein that folds a little bit differently, but in such a way that it's always active. A classic example of this is RAS. RAS is a proto-oncogene that binds GTP. When it's bound to GTP, it's active. When GTP is hydrolyzed to GDP, it's inactive. There are some mutations that prevent the GTP from being hydrolyzed. When that happens, it will always be active, and it will always drive the cell cycle forward. The next way to form a proto-oncogene is through translocations. This could occur when you take a proto-oncogene that is regulated and translocate this gene to another chromosome. And this other chromosome is over here. And the place it gets translocated to is an area that is much more active. Maybe this area is closer to the centromere, where genes are a little less active. And maybe this is further away from the centromere, where it's going to be a little bit more active. So translocating a gene that's usually regulated to an area of another chromosome that is not regulated, and those genes are always on. The next way is through gene amplification. What happens here is you have your 2 alleles that are regulated, and through various mechanisms, these alleles can be duplicated. Maybe it's on a separate chromosome. Maybe it's just placing another copy of it right next to it. But at any rate, what happens is you get more than 2 genes present. And as a result, every time this is activated, instead of just turning on 2, we turn on 4 in this drawing here. So that's all I want to say about proto-oncogenes and oncogenes. I want to move on to tumor suppressor genes next. All right, tumor suppressor genes. We can think of these as the stops of the cell cycle. These genes will make proteins that will hold a cell in G1 until all the DNA has been checked for mutations and either fixed or the cell is targeted for apoptosis. Same thing at G2M. Has all the DNA been copied? Are there mutations? These tumor suppressor genes makes sure that the cell stays at these stops. An example, remember, is our retinoblastoma protein. Remember, it bound to E2F, and as long as it was bound to E2F, that cell was going to stay right here. So again, retinoblastomas hold the cells at these stop sites. Unlike proto-oncogenes, tumor suppressor genes require both alleles to be mutated. To keep everything honest, just in case you learn about this in another class, I'm going to put the word usually. There are cases where they don't both have to be mutated. But for the purpose of this class, we're going to say both of them have to be mutated. So in a healthy cell, these tumor suppressor genes are usually on to stop the cell cycle. But they can be turned off, and when they're turned off, the cell can move past the cell cycle. So they're highly regulated. In a mutated cell, these tumor suppressor genes are not expressed. And it has to be the case for each gene, because if one of them is still expressed, that is usually enough to shut down the cell cycle. But in this case, they're both turned off. It could be a mutation in the promoter that prevents the gene from being transcribed. The gene itself could be mutated. But the end result is we're not making tumor suppressor gene proteins, and so we're not stopping the cell at these cell cycle stops. Somebody who is heterozygous for this, so you might be RB plus, RB minus. These individuals will be able to regulate their cell cycle just fine, but they are more susceptible for cancer. Because unlike this individual, where both copies have to be mutated, if you're in a situation where you have one that's working fine and one that is mutated, in order for this cell cycle to be ignored, you only have to gain one mutation, which is statistically a lot more likely than getting two mutations in the same cell over here. Because of this recessive nature, these heterozygotes, tumor suppressor genes, that is their mutations, can be heritable. If I was heterozygous for the retinoblastoma mutation, I could pass on the mutation to my children. Now these mutations, as I said, these tumor suppressor gene and oncogene mutations can lead to cancer, particularly if you get enough of them, of these mutations. So cancers do require loss of function of these tumor suppressor genes and a gain of function of the oncogenes. However, their impact is greater when the genome is unstable. This genomic instability increases as you go from a benign tumor to a malignant cell. So these advanced tumors here are very often full of chromosomal alterations. Various kinds of aneuploidies where there's more or less total chromosomes, translocations, deletions, chromosomal duplications, inversions. All of these increase as the genomic instability also increases as the cell becomes malignant.